morning, Dr. Tuhina. It's a pleasure to have you here. I'm Charvi Trivedi, and I'm a science communicator at Lab Link Up.
Yeah, and I'm Taslima Farouk. I'm also a science communicator at Lamb Link. Welcome,
Uh,
ma'am.
Welcome, ma'am,
Thank you
to
so much.
Science Unplugged, which is our first ever podcast series.
Yeah,
And so let us dive straight in. Um, welcome, Dr. Tuhina. And Dr. Tuhina is a medicinal chemist at the University of Antwerp in Belgium. And as I said, it's an honor to have you here. Upon reviewing your scientific background, we were impressed by the various amount of research institutes that you've worked in. But we would like to begin with the basics. Can you tell the audience what does the day in the life of a medicinal chemist look like?
okay. Thank you so much for having me here. I'm very, very glad that I could manage with everything and actually present here. So, as you know, that I'm working as a medicinal chemistry scientist. So that means that I use chemistry tools to discover new medicines. So, in the initial phase of my career in this field, the majority of my time, let's say around 90%, it was dedicated for like hands-on bench work in the lab. So my primary task was synthesizing chemical compounds that had some potential to become a new drug. But as I advanced professionally, so my focus gradually shifted away from my lab work to more of like desk-based responsibilities. And as an essential aspect of being a medicinal chemist, it is the integration of chemistry and biology, you know. So over time, I gained experience working in biological setups and laboratories, different kind like biochemistry labs, molecular biology labs, and I was applying my synthesized compounds in biological systems. So as a result, I would say my workday is highly interdisciplinary. You know, a significant of my portion is also spent on collaborating with my colleagues, discussing our research, analyzing the data, and of course, planning the next step to go ahead with the project. And so I would say that it's very difficult to put in a one sentence how my day-to-day life actually I spend. And it totally depends on what point of my project I am right now. Okay, so that completely decides my day-to-day job. But yeah, that's what I do.
What inspired you to take up this field of research, ma'am? Can you tell us a little bit about your journey?
Yes. So I would say that I have always been a researcher from my childhood. And when I say researcher, it doesn't mean like, you know, the high tag or high degree or doing some real life problem handling. Not like that. It's like if you give me a topic, if I like that, I would like to dig deep into it. So that's what I say by a researcher from childhood. So it can be anything. It can be also a pen, you know, or a pencil. And I'd like to know more about it. So I have always been a researcher and I knew that. So that was, I would say that a very big advantage in my early career, meaning during my bachelor degree. But as I grow up with my academia and everything, I realized that yes, this is what I should do. And after my master's degree, when I decided to do a PhD, I was not that much familiar or that much exposed to drug discovery, which I'm doing right now. So I would say it was kind of a coincidence that I got the opportunity to do some cancer research. And once I started, I completely fell in love with it because it is just like any other scientific discipline. It gives you the thrill of discovery, you know, that little problem solving happiness. But what I liked about drug discovery or medicinal chemistry is that I have an idea and I'm actually working day and night to transform that idea into something which can impact the life of so many people. So my work is directly contributing on the global well-being. So I think that is what kept me motivated to continue with this research field. Yeah.
Why Europe, ma'am? Why not India?
Well, I did my till my master's degree. I was back in India, so it's not like I did everything in Europe. So my basic is from India, and I'm very grateful for that. I think we have great capability, and our curriculum is very good for anyone to be a global scientist in any place. Why abroad is or why Europe? I would say that in India we have very limited capacity. Okay, in terms of the number of applicants and the number of good research institutes, and I wanted to spend my time in the lab. Most of my times, it's not like just go to any institute and just do some research, write your thesis, and get a degree. I didn't want to do that. I was very serious. Like if I'm doing a PhD, it should be worth it. And I realized that it's not so difficult to go outside and try to find a good research institute, which is way more difficult in India, because in India we have a setup that you need to clear some exams. In my field back then was like clear NET and GATE and get into the IITs or some CSIR research institutes, but that's it. No other exposures. But when you talk about Europe, there are so many countries, so many research institutes, and the competition is way lower than India. And the next thing, I knew that in Europe, the PhD in chemistry or chemistry related disciplines, it would be covered in 3 years. While in India, it's minimum of 5 years. In USA, it's also the same. In Australia, it's 4 to 5 years. In UK, it's also around 4 years. So Europe was like 3 to 4 years. So that was one thing. And I think when I really decided to sign my contract is because I got a very prestigious fellowship, which is like Marie Curie PhD fellowship. It has only like top, you can say, less than 5% of the people in the world, they are having this fellowship. So when I got it, I didn't have a second thought like which part of the kind of globe it is. So I had to go for it. So that's why Europe. Did you ask something else? I forgot.
That's
No,
so.
thank you, ma'am.
Thank you. That's a very inspiring thought, actually. So I would like to ask a very specific question now.
Yeah.
So what are the current scientific problems that you're trying to address with your research?
Oh, okay. So from my PhD, I would say, because I think that's when I started actually solving some real problem and just not getting some technical skill trainings and just getting knowing the lab life. So I have started doing research in oncology as a drug discovery scientist in oncology. So what I tried to do is working on anti-cancer drug discovery. So in simple words, I'm trying to find a cure for cancer. Yes. And if you want to know into a little bit more details, then of course, so you know, when the cancer cells, they grow in our body, and they do many strange things, of course. But what is most eye-catching is that they become very smart. They're more smarter. They're smarter than the healthy cells, way smarter. So when they develop and they grow, they like to form like a colony, you know, and inside a fort. So if a drug is entering our body, that drug has so many difficulties and so many gates it should actually cross to reach the cancer cell and kill them. So this guard wall is like a resistance mechanism the cancer cells they develop by themselves, and we still don't know how they manage to do it. So your drug might be very good at killing them, but first you have the drug should reach the cells, right? So what I'm trying to do is making this guard wall weaker, and for that I'm targeting many cellular mechanisms which we think that the cancer cells they're using to make this guard wall. And also sometimes what we see is that even if the drug is reaching the cancer cells, the cells they just send some signals and tell that this is an illicit substance to our body. So once it is orally taken, every machinery started being active, destroy it, and it should be excreted out from your body. So I'm trying to see how can I make cancer cells a little bit more stupid so that my drugs actually can do their job, you know? Yeah, so that's the thing I'm doing.
That's actually a very interesting aspect to cancer cells. Another thing which I would like to ask is obviously your job sounds very challenging, and how do you come across any hurdles that you face in your day-to-day life with respect to
So
your
the
work?
thing is that there are people who are working on anti-cancer drug discovery for a long time. So we already know that the success rate is very low. So we already know that. So when you start with this kind of mindset that it's full of difficulties, it's not an easy job. And for any drug to come into the market, it's like minimum 10 to 15 years of work. So when you start your PhD and you know that you have only 3 years of doing it, you already have started your journey by convincing yourself that you're not going to discover a drug which is going to be in the market before you finish your PhD. You know that's not the case. You know that's not the reality. And it's a highly challenged and interdisciplinary work. So I would say that I knew that what are the obstacles coming into my way. But I also kind of focused on the positive things, positive outcomes I was getting. And in my mind, there were two things very important, two factors, I would say. First one is to finish my PhD and get my degree, because of course I was on a visa, the fellowship, it has some time, it should expire. So that was my first priority. And second is that how I can upskill myself in every day. It doesn't matter if it is a success or it's a failure, but you have to learn from everything. So every day was a learning experience for me. And I think I was more focused on that. I didn't focus like how many publications I'm getting, my colleague is getting 10, why my number is 1. No, that was not my point of bother any time. No, I was not bothered at all. So I knew that every day I can learn. So I was focused more on my growth instead of comparing myself with other people, instead of thinking that, oh my god, that people who are in MIT, they are doing so great chemistry or great drug discovery, I don't have that much scope. It's okay. So I kind of accepted these kind of limitations before even starting my journey. So I think that helps me a lot to survive. Yeah.
It's very inspiring of you, ma'am.
Some noise issue.
More about this
Yeah.
drug discovery.
Taslima, can you repeat your question? Oh,
Yeah.
Can you hear me?
Yes.
Am I audible
You
right
can-
now? Okay.
Yes.
Ma'am, whatever you have said, it sounds so inspiring to us to know that you put yourself above everyone else. We would like to know more about drug discovery also. Like building something therapeutic from scratch sounds just amazing, because I'm also from the same background and it is
Yeah.
really inspiring to me. So how do you
Yes.
come up with a lead molecule for each project? Is it always trial and error, or is there one-shot successes as well?
No, it's never a one-shot success. It's never. It's always trial and error. It might take you 5 to 10 years to just find your lead compound, and that is completely normal. So, what I would say that in drug discovery, how we try to proceed as a medicinal chemist perspective is that first we try to find a target. Let's say my background is in cancer, so of course I would try. Just let's focus on breast cancer. Okay, so I would try to see like the people, the target identification people, which means like they can be some kind of structural biologist, they can be some even computational chemist who are doing bioinformatics, or people who are reading proteins, so they try to there are these people who go to the patient of the breast cancer who are in the hospital going under treatment and try to extract some cells, from the cancer itself, the cancer body. And then those cells, that goes into the lab. And then people try to study those cells, like what is the problem, why they become cancerous. And we try to see which kind of phenotype the cells they have actually adapted. That's why say they become so malignant to our body. And let's say we find that some factors, some proteins, they are not working because our protein is our building block. Each protein, they have their own role to play. So maybe one protein is not behaving well, and then we try to see, is it the same thing in all the patients? Is it like a common factor, or it's just to this patient? So once we find one factor which is almost 80% relevant to all the breast cancer patients, then we can think that, yeah, this protein is something we should read more about, we should do more research about, and we should try to think and try to find why the case, why it is not behaving the way it should. So once we find this protein, we call this as a target protein for treatment of anything. Let's say here we are talking about breast cancer. Then what we try to do, go back into our basics. Basics means school level basics biology. So we know that all the proteins, or let's say enzymes, they like to follow this lock and key method. So each protein, they have a keyhole, and then there should go like a special key with a special structures which will fit to that keyhole, and then the protein or the key would be opened. So this is very simple. So it's the same way, like just like lock and key, each protein has their active site, which is the keyhole. And then what we try to do, we try to target this active site, either to close it so that the protein is not working, or either to open it so that protein is working. Okay, depending on the on the kind of lookout we are focusing on. So once we find the active site, we try to characterize it, we try to read it with the kind of amino acid residues it has, and then we try to design compounds which can actually have some interaction with these active site amino acid residues. Because one thing we need to understand while designing this kind of compounds is that inside our body, there is no light, so when you take a drug and you have a pain on your arm or something, how do you expect that the drug will actually reach to the point and actually react with something so that your pain is getting minimized, or anything, your wound is getting better? How does this happen? Because there is no one in your body which is targeting, oh, you came, now you go there. No, right? It doesn't happen. So the drug, it kind of flows in your body with the blood vessels and with the other stuffs we have inside our body, and then it's getting hit to another one protein and says, no, 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 I'm not fitting there. So the protein will kind of push it away, that go away, this is not your place. So it's a very difficult method and very complicated mechanism how a drug actually reaches to your target protein at a specific organ of your body. So that's why it takes so much time to find a compound which we can say that it's a lead compound for a specific protein, for a specific disease. Because let's say I have a target protein A. Now that target protein A may be presented for the breast cancer people on their breast, but that protein might be also present in the liver or in the heart. So if we stop that protein from working, we have to make sure it has been stopped only in the breast, not in other organs. And that's why cancer has so many side effects, because the drugs, they get crazy, they go everywhere in our organs, and we start having the side effects. And we know this is one of the very big limitations. So in drug discovery, we say it as the selectivity. So your drug should be very selective towards the active site and towards the active organ. So that's why the lead generation is not a joke. It takes long time. And once you find a good lead compound, then of course there are the next steps. Also, even if you find a lead compound, doesn't mean that it's a drug. There's so many steps,
That's very insightful. So are there any unusual experiments which you might have done in your lab, or any unusual observations that you got and which you thought or think can be a potential breakthrough in drug discovery?
Yes. So I would say like the anti-cancer drug discovery, which I'm working on right now. As I told you that we are working on this phenomena of reducing the drug resistance mechanism by the cancer cells. So we all know that there is something called autophagy. So autophagy is like self-eating. So it is very important for our body to maintain a cellular homeostasis, to maintain the body healthy. If the cells are old enough, they are getting died. And then if there is anything component like amino acid residues which can be used as a future food, then it is also getting stored in somewhere. So it's a very well communicated theory. And we were trying to focus on this machinery because the cancer cells, they use this because they are very hungry. They are always growing, so they are hungry. So they take food from the healthy cells. That's why we intend to lose weight, the cancer patients, because the food is going only to the cancer site and not getting distributed. So we wanted them to starve, the cancer cells, that no, this is not happening. You are not getting the food. So we wanted to do this. But then what we saw is that we are dealing with cancer treatment. So it's good if your cancer cells are getting weak, if your cancer cells, as I said, getting stupid. But that's not the ultimate goal. The ultimate goal for the cancer is to kill them, is to get rid of it out of your body, right? So every time someone is focusing on drug resistance mechanism, they need to do kind of co-treatment therapy. So they're actually injecting two drugs at the same time. They're taking two drugs at the same time. One is killing the cells, another one is reducing this resistance mechanism. So we thought, why two drugs and why two side effects? Each drug has their own side effect. We need to think that the patients, they don't like to take so many tablets, the lower the better. So we wanted to minimize this number of injections or oral drugs, minimize the amount, the dose, So we tried to think that why not one drug designed in a such way that it can have two hands. One hand will kill the cancer cells and another hand will help the drug to get through this fort and break the guard wall and actually reach to the cancer cells. So our plan was to design one drug with two different mode of actions. So I think that is very interesting. It should be very interesting to the people who are working in anti-cancer drug discovery, and now people have started working on this. So I'm hopeful. Let's say where it goes, but I'm very hopeful about it.
That sounds really interesting, ma'am. But what was the hardest setback you have experienced during this process?
So I would talk about this particular project where we were trying to design compounds with two mode of action. So the problem was the compounds were very complicated. They're structurally very different to synthesize, very difficult to synthesize. That was one thing. And they have like some 3 ring arrangements which were not that much stable. You need to think that when you take a drug inside your body, it plays with the pharmacology, because it gets the acidic environment in your stomach, and then it has to get passed through liver and everything before it reach the target. So the chemical stability was a huge problem for our compound, the kind of compound we were designing. But I would still say that the most difficult thing was synthesizing the compound. So in the first 2 years of my PhD, I synthesized only 5 to 6 new compounds, which is like very bad statistics. That's why I had to follow 2 projects simultaneously. One was my main project, but we had to be prepared, like if this doesn't work, then I should switch to the other project because I need to get the degree and I need to give my report to the scientific committee which are reviewing my work. But guess what? I spent so much time. My students, they spend so much time, because every time I had a student, I'll start giving them this problem, that this is the synthetic problem I want to solve and I'm not able to solve it. So can you guys please look into it and just tell me what you want to do? It was the last 4 months of my PhD thesis when I had a student, and when we found a new reaction. It's not a new reaction we found. The reaction was already there, but we couldn't see that that could be used with our compound as well. And we could use that. And when we did the reaction, the pilot reaction, it worked. We were so happy. Like the whole lab was celebrating that Tuhina got that reaction worked, you know? So then I'd say I finished my PhD with 21 new compounds, and that happened at the last stage of my thesis. So that is the most difficult thing I was dealing with, because everyone was so worried that I'm not getting new compounds from my project. I'm just working on Plan B project. So I think that was the most difficult thing I dealt with, but I'm very happy that I came out of it like with a positive outcome. But here, which is very important, I would like to share that you have to give the students, like the master students or the bachelor students who are coming into your lab, you have to give them the liberty. Because after 2.5 years, my mind was set in a certain way that I would look into some specific points, because I'm thinking that I have looked into everything. I have checked everything. There is nothing else we can do. But when a student is coming, they're coming with fresh mind. They'll come with stupidity. That stupidity might be helpful for you because you can make that stupid concept in a smart concept and apply it. But maybe you never saw that stupidity, because you are so focused on so many things. Sometimes you kind of overlook this kind of small information. And it happened to me twice in my PhD. So I would say to every people, like, never underestimate the power of new people in the lab.
I think
What kept you the best?
yeah, I would just like to add something that I can
Yes.
actually relate with what you said right now about I mean, being a master student myself, I had the liberty to carry out certain aspects of the project myself, and that really helped a lot in our work.
Yes.
So
Yes. Yeah, that is
so
true. That's true.
so
What was your best motivation, ma'am, during this difficult time for you?
Sorry, I didn't get you. Can you please repeat?
Ma'am, what was your best motivation during this difficult time?
Best motivation. I It's very difficult to say what was my best motivation, because I never, as I said, I was never focused on the publications or the outcomes I was actually getting. I was more of like enjoying my journey. And I knew that I would do my PhD here, and then I'll move for postdoc. I already have the labs in my mind, the lists were ready. So I was already focused on the next chapter. So even if my present things are not working that the way I want, I knew that I have to wrap it up and move to the next chapter. So I think I'm always looking for my future, and that is the best motivation. Yes, and another thing I would say that if you look back into my journey, the place I belong from, like where I started and where I am today now, then dreaming beyond my capability and beyond what I see is the thing which made me the person I'm today. So I'm never scared, or I have any fear of dreaming big. So I can totally ignore the problems which is going on right now, and I can focus like what I'm going to do in my next 2 years. So I would say that my life has kind of engineered myself in that way.
I think whatever you just said right now, it is completely to the point, and it will be the highlight of this talk today.
Yes, because I forgot to mention this. I think you guys have asked about sharing my journey. So, I belong from a very rural area of India. You know, it's the mangrove area. It's like during my childhood we didn't have electricity, we didn't have the roads in my village, so it was like the mud made road. And like I used to go to the schools with the bags, and then the dress very somewhere it should be stuck so that it don't get the mud. Once I reach the school, I need to clean myself. Sometimes we just used to go to the pond and then also washing our dress. So that's the kind of background I am from. We used to walk 30 minutes every day to collect the drinking water. And so that's the kind of background. And there was only one school in my area, and it was again like 30 minutes walk every day, and so in total 1 hour back and forth. And it was a Bengali medium schools. The schools didn't have enough teachers, so teachers were basically part-time teachers who were coming just for 2-3 months, and then again another new teacher. So I kind of had to self-study myself. And I didn't have the privilege to go to tuition. We were having financial issues in my family back then. So I started, I didn't start from zero, but I started from negative. So but I I always knew that I have to go out of this place, because I like to read from my childhood. And I was reading a lot the novels, the journals, the magazines, everything, even the film magazines. I was reading a lot, and I knew that the world I'm seeing, the world I'm experiencing right now, that's not it. There is more thing, you know. I wanted to explore. So from the time I was in my sense, I knew that I had to go to Calcutta for my bachelor degree. Otherwise, my life is finished. Because in my place, no girl, they go beyond 10th standard. They get married off, they have children, and they need to start their family. And then my family, my background, so they are Muslim community. So in my place, it's even worse for the Muslim people. And everything is a restrictions for them. Forget about even going school. It's about everything, the way you lead your life, the way you dress up. So everything would be questioned, at least in my place. So that's the kind of background I belong. But I always knew, as I said, that I have to get out of here. It's not because I hate this place, but I know that I'm capable. And I knew that I would be able to make it. And I wanted to break that chain, which my clan or my family were bearing generation after generation. So I'm the first girl of my family who went for a bachelor degree, who studied science, who did master's degree. Forget about PhD. No one in my clan, men, women, no one did it before me. Even in my whole area, I was the first master degree or bachelor degree holder woman. So people were so surprised when I was doing this, but I knew kind of, because I met some great people in my life who kind of motivated me. I used to read stories, and I knew that it was possible. So yeah, that was my motivation. And I think my journey is very interesting from my point of view, you know.
It is. Personally, I find it really inspiring. I can actually relate to it a little bit. So,
Yeah, it's indeed very inspirational.
Yes.
Our listeners would like to know, what is the future scope in your field of research currently?
Okay. So as I said that I'm into drug discovery. So basically I'm talking about the biotech sector. And in the biotech sector, it's a huge market right now, you know. So and the market is continuously growing. They say that there are two professions where you should never be out of your job. One is health, because people will get sick. Another is law, because people will have lawsuits. People tend to fight with each other. So the biotech sector is growing, and it will grow because the way we are leading our lives, it's becoming more complicated each day. Even if we find a cure for cancer tomorrow, it's not the end, because the day after tomorrow, the way we lead our lifestyle will have something more dangerous and more complicated than cancer. So the market is growing. Biotech is never going to stop. That's the first thing. So it's a very good starting point. And second is that if you have some drug discovery research experience, you can, of course, go into the R&D sector of the biotech. And in the R&D, you already know that mainly you will do the discovery research or some kind of product development and or clinical development, let's say. But there are other things you can also do. For example, you can move into the commercial side of the biotech, because you always need to remember that it is easier for the scientist to train them for business than the reverse. So we scientists, we already know the science. We just need to understand that point, like how we can monetize the scientific knowledge we have. That's why if you look into the founders or the co-founders, CEOs of the big pharmas, if they are getting it because they have their family business, then it's different. But if you see the inspiring figures, they all have science background, because it also helps. If you want to invest into a new project, if you're trying to pitch a new proposal, a new scientific proposal, then you have to consider what are the limitations you might encounter while leading the project. So if you don't have that background, you won't be able to do it, right? That's why you have a huge scope to move into the commercial side. And very easily, I would say it's very easy to move into the commercial side, where you can, let's say, you can work for the marketing team, you can work for the sales team, you can work for the corporate communication. Because even the big pharmas, when they have a drug, they need to reach to the cancer, let's say the general practitioners, the cancer treatment hospitals, and actually tell them that our drug is good, we have this. So you have to kind of recommend the drug to the patients. So these are all very linked to your drug discovery profile. If you don't know the structure of your drug, why your drug is good, what is the selectivity, what is the success rate of the clinical trial, how you are going to pitch your drug. So you can be also into the cell teams, you can be in the marketing team, you can be in the business development team, and there are so many other things you can do. While it's like you are like growing, and you are getting some new skills by using the skills you already have, you know yes. So also there is the possibility to move into manufacturing, quality control, and you can be also into the recruiting team. So provide some service, because what are the kind of scientists you need for your company. So it's a huge plethora of like options. Once you are into the field for 5 to 10 years, you can really see the best option you should opt for. So it gives you lots of opportunities.
As a scientist, what advices do you have for young, inspiring scientists for the next generation?
I would say that, as everyone said, dream big and have high hopes. Don't think that you cannot do it because someone said you cannot. And I would say one thing, especially for the Indian setup, because I belong from there, so I know the mentality. It's that cracking the NEET, cracking the IIT JEE, going into IITs, it's not the end of the life, or it's not the beginning of your life. No, that's not true. If you do it, it's very good. If you can't, there are other options. See, you have to go for that. Every year there is a Nobel Prize for doing research in science. How many doctors got the Nobel Prize? How many engineers who topped in your class got the Nobel Prize? Think about that. So don't look down upon the people who are studying basic science, because they are going to contribute in the global health, in the global problems, in such a way which is impossible for a doctor, for an MBBS doctor. I'm not degrading anyone. I'm not doing that. I'm just saying that everyone has their role to play. No one is superior to another. So if you don't get a chance, it's fine. There are other chances. So go and grab them. Okay. So But we have this mentality of saying that highest grade is the best. No, that's not true. As I said, for if you want to be a researcher, you need that mindset of to be a researcher, which means you need to stay realistic. You need to be built in such a way that you can take this kind of failure, everyday failure in your lab job. Even you are working for it for 4 or 5 years and you are not getting any result, are you able to handle that? Only then go for research. Otherwise, don't go into research because you are the topper of your class. No, that doesn't work. So there are people in our ISC Bangalore, which is very prestigious, which is the number one in India, and there are people, good people, qualified people, and they have capability and enough capacity, and they're there for 10 years, and still their PhD is not over. So something somewhere went wrong, something didn't work. So it doesn't give you the certificate that you will be a good researcher because you have been scoring good. I mean, if you are scoring good and you want to be a researcher, it's very good. It's very good. But to be a researcher, your grade, it doesn't matter. That's how I see. Because theoretical knowledge and practical applicability is two different things. You can be very good in theory, but you cannot transform that theory into your hands-on experience. So that's why I would say that if you really like doing research, then go for it. And there are so many options in India, so many options outside India. So just you need to keep trying.
Yeah, that's something very good said by you. So with that, we come to an end of our podcast. Thank you so much for your time, and your words will surely leave a great impact on our listeners.
Thank you so much.